Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius podcast series. This is the Health, Medicine, and Bioscience edition. My job is to find what I call the geniuses, the 0.1% of people in their fields. I've spoken to over 2,000 scientists, clinicians, researchers, etc. And today I have Roy Black. He's an affiliate professor currently with the University of Washington in the chemistry department. And Roy has a long career in biotechnology. He built and led R&D programs that identified enzymes required for the release from cells of two central mediators of inflammation, which are pretty popular or have been talked about a lot, which is interleukin-1 beta and tumor necrosis factor alpha. So we're going to talk a little bit today about a little bit different subject, but origin of life and how that may have occurred. So Roy, thanks for coming.
Thank you.
Yeah. So why study the origin of life? Is it a fascination for you, or how did this come about?
Right. It was a bit of an odd migration from the pharmaceutical industry. I've always been curious about how the world got to be the way it is, and I actually majored in history when I was in college. But a lot happened before recorded history started. After college, I did some reading in anthropology, but eventually I worked my way all the way back to the ultimate question of how life itself began. Then more proximally, I read a number of books about it in the mid-2000s while I was still in the pharmaceutical industry. And I was really shocked that there weren't any comprehensive answers. And even the partial answers were not compelling. So I started sketching out ideas myself in a notebook and got some schemes that I thought were worthwhile. I did a reality check with a couple of thought leaders in the field, and they were encouraging. So then I before leaving the pharmaceutical industry, I did a sabbatical to kind of check out the field, kind of a reality check, and then left the pharmaceutical industry. And so that was 9 years ago.
Okay, so is there commercial interest in the origin of life, or is this more for for people's I don't know if you say gratification, but just for knowledge in general? How do you see it?
Yeah, I think the main benefit of a better understanding of the origin of life is it helps to satisfy our curiosity about where we came from. I think it brings home the long journey to where we are now. It's a long way from the simple theory I'll sketch out to cells that extract energy from the environment, to multicellular organisms that think. So to me, understanding how all this started from a very simple beginning deepens our appreciation for life as we know it and for being here. Those are my motivations and what I see the payoff. Well, one other point is that it will help to answer whether there's, or at least the probability of life existing elsewhere in the universe. And whether that has any other than curiosity payoffs is up to each individual. But
what are your thoughts there for a start? Do you think that it's possible, probable, or there must be life in other parts of the universe?
Yeah, well, as we'll get to in more detail, I think our results increase the probability somewhat. They show that bringing the pieces of a cell together is not just a rare random event, but actually occurs through a natural affinity among the molecules involved. So in that sense, I would say finding life elsewhere more probable, but still that long road that I alluded to a few minutes ago That was a 3 billion year road. We think the first cells arose probably about 3.5 billion. So even if simple cells do exist elsewhere, I'm not confident that they followed that long road. Complex thinking organism.
So you think is it likely that there is life on other planets, but it's not at an advanced stage, or what is your thought there?
I'm frankly skeptical. I think you need the right ingredients, and in the right conditions, which again we'll talk about a bit more further on. The answer to that that you frequently hear is yes, but there are billions and billions and billions of planets out there. So even if the proper conditions are rare, they're out there. But I'm skeptical that it's
Okay. So all right, well, for the origin of life here on Earth, now in summation, what do you currently believe happened? How do you think it happened?
Okay. Well, our scheme in a nutshell is we start with the question of what is life. Okay. And in our view, all life on Earth is composed of cells, and cells are fluid-filled sacs that can grow and divide, and that contain two additional major components, RNA and protein. And just two additional bits of background for those not up on their biology. The RNA and protein each are composed of subunits, or what we call building blocks, strung together like beads on a string. So that's our view. Life is composed of cells. Cells consist of these fluid-filled sacs containing RNA and protein. So the problem, as we see it, is how did cells arise on the ancient Earth? And we think there are two major problems. First, how did the pieces I just described get together? And then second, how did the initial aggregates persist in the face of a challenging environment? So briefly, our answer to the first problem is that there were molecules on the early Earth that formed sacs spontaneously. That's been established. And that the building blocks of protein and RNA stuck to the sacs. That's the simple heart of our concept. So once all the pieces were brought together in this way, the reactions between the protein building and the RNA building blocks could have proceeded to yield RNA and protein. So what about,
yeah. Here, so this kind of event, I mean, it seems unlikely. If it happened once, that would be enough for it to continue. So, my guess is that it would have, if this is what happened, it would have happened millions, billions, maybe trillions of times. But how do you go from RNA and protein sticking to, the inside of a lipid vesicle, let's say, to this entity now replicating itself? How would there be any knowledge or desire for something to replicate itself?
Right. So there is a second component to our scheme, and that is that these initial aggregates, as I said, would have been unstable in the face of environmental challenges. We know that the kinds of vesicle or sac forming molecules that were present on the early Earth form relatively unstable sacs. So, for example, they're destabilized by high concentrations of salt. So our answer to this second problem is that the protein and RNA building blocks that stuck stabilized the primitive sacs. So the result would be a self-reinforcing mechanism. The more building blocks bound, the more stable vesicles there would be for further binding of building blocks. And so the drive for complexity that you're referring to was initially a drive for stabilization of initial aggregates. So any changes that occurred that would increase stability, such as joining the building blocks together to form RNA. If those changes increased the stability of these initial aggregates, they would have been preserved, and those sacs or early cells would have outcompeted others. So that's how you get the drive toward complexity. Of course, there are many steps along the road. Again, as I referred to the long road, eventually you needed the evolution of proteins or RNAs that extract energy from the environment, that catalyze the synthesis of new building blocks. But we think all that's possible once you get this initial population with RNA and protein going. And an important point that one of my mentors, David Deemer, likes to point out is in a single teaspoon full of water, there can be millions of these little sacs, okay? And there are billions and billions of sacs worth of water in the ocean. So you were alluding to this, there were many, many, many shots on goal to get combinations of these early form molecules that had function. And again, the early functions would have just been for stability, growth, and division of the sacs.
But have you done like a Urey Miller type experiment where you tried to make the conditions right and formed a bunch of these sacs in a tank or an environment and see if you can get them to take the next critical step?
Right. Well, so yeah, let's talk about what we have done. We have done real experiments. We have published papers, and yes, we have shown that both RNA and protein building blocks will stick to or bind to very simple what we call vesicles, membrane sacs, all synonyms. And that was published in two prominent PNAS papers, one just last summer. So yes, that happens, and which I think surprised a lot of people, or people hadn't actually asked the question. And we showed that when these the building blocks bind, they do in fact stabilize the vessel against high salt concentration or precipitation by divalent cations. So that's the experimental basis that we've established. Now, getting further down the road to your question, indeed, where we're going, what we're trying to do now is to show, first of all, that when the building blocks stick to these vesicles, that actually does facilitate their joining into RNA and protein. And if that's true, that would be a huge advance in the field itself, because it's still really not well understood how the building blocks, either a protein or RNA, got strung together in the early environment. So if our binding to our sacs facilitates that phenomena, that's a big step forward. And we're working hard to answer that question. Then the second big question that we still have to address is, if that happens, if the sacs do facilitate the formation of RNA and protein, do short stretches of RNA or protein further stabilize the sacs? In other words, does joining the building blocks more effectively stabilize the sacs or increase the rate of growth and division than unjoined building blocks do? And if that's true, then you really have the beginning of Darwinian evolution. You have RNAs and proteins that are selected, and in this case, the initial selection is for again, stabilization and increasing growth and division of the vesicles.
But how do you go from, again, even if you see stabilization at the level of short, spans of RNA, you still need to then make the next leap to replication, faithful replication of the same thing that exists, that divides and replicates. How do you get to that step?
Right.
What would be the driving force? What would be the driving force for that? I would think that would definitely have a hill of instability that it had to overcome to divide and then, reach the new state.
Right. So that transition to anything like what we have in modern biology is really a black box in anybody's concept. But for example, one way to get there is if you have even just a short stretch of protein building blocks, amino acids. Most of our listeners do have that much biology, but so a short stretch of amino acids strung together, if it catalyzed its own formation, that would be the beginning. And that's not implausible. It may sound implausible at first, but actually people have shown that just two amino acids joined together, a dipeptide, does have significant catalytic activity. A simple dipeptide can catalyze the formation of sugars from simpler molecules. So again, this would be a rare dipeptide that could catalyze its own formation. But again, we're talking about billions and billions of shots on goal. So that would be the way it could get going. But yes, ultimately, you have to get this interaction between protein and RNA that you see today going to really generate complexity that is preserved. As you say, you need a hereditary mechanism. So what I just sketched out is a very, very simple one. But how do you get to the complex version that we see today? And we have to envision that once you've got proteins and RNAs, again, in the same place, which I think is a critical issue most folks in the field don't address, you've got them in the same place, there's the opportunity for them to start interacting together and catalyzing each other's synthesis, which is, exactly what happened today.
But what's the probability of the conditions being right? From what I've read and seen, it's just low probabilities stacked upon lower probabilities stacked upon more and more and more. So it just seems so improbable that the conditions would be right and multiple factors would be right to create life. But I mean, do you see it that way or no? Or do you think it's highly probable?
Well, I said, going back to the beginning, I'm skeptical whether it's happened elsewhere in the universe. Yeah, I think you need to get these essential building blocks made. That's not considered a problem. You actually find the building blocks on meteorites. And a lot of work in the early days, going back to the 50s and 60s, showed that they can be made by abiotic processes plausibly occurred on the Earth. So the existence of the building blocks is not a mystery. There's the question of whether they would have been present in a high enough concentration to go on to more complex forms. But that's where one of the strengths of our concept comes into play. Again, the sacs do form spontaneously. You need some significant concentration of the lipids, as you said, but that part's not mysterious. And then if our simple sacs, which would have formed spontaneously, bring together the other pieces, it begins to look more plausible. One other important point is that there's currently a lot of interest in the idea that all this took place in pools of water on land that would have been subject to drying and wetting cycles. And during the drying phase, that would have concentrated all these components, making, again, their joining into more complex structure, more plausible. I think that's a big advance over what has been a popular idea that all this happened in hydrothermal vents in the ocean. We can discuss that if you want to. But I think that this idea of pools that could have dried down and concentrated the ingredients definitely increase plausibility. But again, now you're adding another requirement, right? You need land, which there's debate about that. When initially it's thought that the whole Earth was covered by ocean, and so when did land actually appear, and what's the likelihood of that having happened at the right time?
Yeah, that's true. When is current science? When what does it say? What time period did the oceans recede or dry up enough where land appeared, or was land
Right.
thrust upward through the ocean? Like what's the? Yeah, it's kind of weird.
Right, it is, and this is actually one of the really hot areas in the field where I think people are the geochemists and the planetary scientists are really focusing on this now and trying to come up with more realistic assessments. But there's a current view that all this actually could have happened about 4 billion years ago, somewhat earlier than previously thought. There was another issue that kind of dogged an early start for a long time was this idea of a late heavy bombardment of meteorites, which is yet another complicating issue. If that actually happened, it was thought that would have sterilized the Earth, eliminated any life that had guns. So now the planetary scientists, maybe there really wasn't a late heavy bombardment after all, and the configuration of all the pieces could have been about. Actually, they're saying 4.1 billion. Though some people will laugh at precision, but that's the idea. That also connected with that is going back to your question about the Urey-Miller experiments. It's thought that you really needed what's called a reducing atmosphere for these reactions to form the building blocks abiotically. And so essentially that means there would have had to have been some ammonia in the atmosphere. And for a long time, that was thought to be plausible. And people, to some extent, actually disregarded the Urey-Miller results for a long time. They said, they used an unreal atmosphere.
Okay. Quick question here: Do you see what I guess science was called natural selection acting upon non-living things or systems, or just living things?
What I'm proposing is a unit that, you could say was not yet living. These simple sacks just bound the building blocks. I think selection goes all the way back to those, and I think that's part of the power of our concept, because you have to get selection going at an early stage, as you're inferring, to really progress.
I mean, but like even today, do you think natural selection acts only on living things or non-living things as well?
Well, natural selection, as we typically define it, Darwinian selection, a population of entities with variants in their traits and with hereditary components so they can pass their traits on to descendants. I don't see that in the non-living world today, but of course the world today has been taken over by living entities, and so again, we're proposing that this could have happened early on, before there was any life to eat up such a thing.
The reason I ask is why at some point in the past would there not be this force of natural selection, and then all of a sudden it would be there and acting upon this early, prototypical life arrangement? Why all of a sudden would this force then act upon that when it didn't before, when it doesn't now on non-life?
Right. Well, again, it doesn't now because there aren't many or any free building blocks or free lipids, sac-forming molecules floating around to be acted upon by a selective, because living creatures devour them. The bacteria are very good at that. So that's why it's not happening now. That question comes up. Sometimes the creationists will bring this up. Well, why doesn't life start now then, from scratch, if it ever could have? And that's the simple answer. That you don't have these ingredients accumulating like they would have before there was life to eat them up.
Well, why is a certain arrangement of molecules unique in such a way that now, I'm not saying it'll become life, but why would this arrangement of molecules that we call, let's say, RNA or lipids plus RNA plus proteins, why that would then be subject to this new force of natural selection or replication or all these things? Like, why that arrangement? What's special about it that would all of a sudden enable a new force to act upon it?
Well, you use the term all of a sudden. Remember, this whole process probably took place over millions of years. There was plenty of time. We're talking about a process that began 3.5 to 4 billion years ago. And if it took a few million to get started, that wouldn't be all surprising. But I think the critical point is that there was a time when these critical ingredients I'm describing were present, brought in by meteorites and or formed by abiotic processes. They were able to build up significant concentrations because there wasn't anything like bacteria today. And these initial structures formed spontaneously. That, again, is really the heart of our proposal. The idea that the sacs would have formed spontaneously is, again, that's easy to show. I mean, you just take lipids. This is kind of like biochemistry 101. Take most lipids and just mix them. Water, they will form enclosed membrane structures. And the part we're bringing to the story is that the additional components required actually have an affinity for these simple sacs. So that's what was different. There was a time when these pieces accumulated, as that's not improbable. Once there was, we're past the late heavy bombardment that ever existed, these pieces would have accumulated and would have started interacting by these natural affinities. But you had to have it. Sorry, but I mean, just to elaborate a bit, you needed the right conditions in terms of the pH, temperature, salts. Again, couldn't have been too high, but all that's reasonably probable.
Well, today, I mean, life exists obviously everywhere, but when people ask you why can't life start today, well, okay, maybe the conditions aren't right for it to start. So the conditions are right, though, for life to perpetuate itself now that it's here, which is pretty interesting and unique. So is that what do you agree with that, or there's life from non-life?
No, right, life. It had to evolve to the point. I mean, the initial idea, the idea is that initially, as I said, these ingredients accumulated because there wasn't any to eat them up. But at some point, these early cells that I described would have absorbed all the pieces that were available. So then you had to go through a stage where the cells evolved a way to extract energy from the environment to make their own building blocks. So clearly that was a critical step in the process.
But does this mean that there was one window, whether the window is 100 million years wide or, however wide, in which life could start, but that window is now closed from that point? I mean, do you think life started only once in a given window and then never again?
Well, I think I think that's probably true, because fairly quickly, once you got these little sacs absorbing out, pulling out the building blocks, that there wouldn't be the ingredients left for a second start. So if you wiped out life now, of course, all of the existing life would decay back to the building blocks. So yeah, it's an interesting question. Then at that point, could it all get started again? Sure. In theory, you'd have a gigantic pile of these building blocks, because again, along the way, life evolved a way to make more building blocks rather than just relying on what had been brought down by meteor made by eye. So there would be a gigantic pile of these pieces to start reforming. It could start again. That's an question. People like to say that if you just take a cell and bust it up, it's not going to reform. and maybe this is what you're holding it up as a real challenge to how it ever could have started to begin with. But yeah, give me a few million years and maybe it would. It would have to go through these early stages. No, it wouldn't form a full-blown modern cell, eukaryotic cell with mitochondria and so forth. But
why, if we look just at the past 4 billion years only, on Earth only, you think that life started only one small subset window of that time? Why wouldn't it have started again, let's say 3 billion years ago? And then there's two parallel tracks of life.
Right.
Why couldn't that happen? Why? Even if there was a buildup of these proto structures and all that, then all of a sudden, all right, life could start. But why never again?
Yeah, it could have. Yeah, I mean, that's a good question. There's one view of the field is that before the so-called last universal common ancestor of all life on Earth, there could have been multiple starts. I think the simple answer is that the form that we see today, this really striking and not at all obvious combination of lipid, RNA, and protein, once that got going, it would have just taken over any other forms that got started, and as I said, used up all the ingredients that were available. So there could have been multiple starts early on before the last LUCA, the last common, but the last universal common ancestor was so efficient at utilizing what was available in the environment that it simply outcompeted any other forms that had started.
Well, at this moment now, I mean, we could say that we've outcompeted everything else, but then again, I don't even know how many bacteria in the world, how many viruses, how many fungi, so
right.
We've outcompeted maybe in one way, but in another way, maybe we haven't. They're still here. There's still plenty of them.
You're right. Well, yeah, they're all our brothers and sisters, so to speak, in the sense that we are all related. We all descended from a common ancestor. Our biochemistry and molecular biology is incredibly similar to bacteria and so forth. So yeah, there's competition among these forms of life, but it's really one form of life. Yeah, I think if we keep going the way we are, climate change, humans may wipe out themselves, and so then the bacteria will be left, and they'll take over. But I wouldn't view that as a new start to life. As I said, bacteria.
Yeah, I mean, not only have we not outcompeted them, but they are us. We're all holobionts.
Yeah, and you could have a whole podcast on that you have about how more and more in the last 10 years we're appreciating to what extent we are actually composed of that.
So what? I guess last question or two. What is the future of life, or is that not a concern of yours? I mean, you just want to figure out the origin, which is plenty good enough. But
yeah,
do you have thoughts on the future of it?
Yeah, that's keeping us busy. Yeah, I hadn't actually thought that much about it. I think it really does. Well, we know that eventually the Earth, of course, will end, and so I guess at that point, if there isn't life elsewhere in the universe, that would be the end of it. What happens between now and then? Well, the geneticists say that humans are actually still evolving. So, and then of course, there's a lot of buzz about our interaction with artificial intelligence and so forth. And, a lot of people will say that's where we're headed to more of a mix of machine and biological life. But I'll have to leave that to the technologist futurists. It's a bit beyond my pay grade.
Well, very good. Well, Roy, what's the best way for people to find out more about what you're working on?
My co-investigator is Sarah Keller, and she maintains our website at the University of Washington. So that would be this, or just Google either her or me. We have these published papers that people can get started.
Well, very good. Roy, thanks for coming on the podcast. I appreciate it.
Okay. Thank you, Richard.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.